For decades, scientists tried to reverse engineer human history. By analyzing the genetic differences between people alive today, they attempted to trace lineages backward, hoping to map how our ancestors first spread across the globe. The operating assumption was that human expansion was a tidy process. The models of that era suggested that as early humans left Africa, they fanned out in distinct, isolated branches, replacing older groups without significant blending. But reconstructing the deep past using modern DNA is like finding ancient footpaths on a modern highway map. Over millennia, humans have moved and mixed so frequently that recent migrations have essentially overwritten older genetic patterns. The original signals are often blurred or erased. Relying exclusively on people living today created a false impression. It made human populations appear far more isolated and pure in their evolution than they actually were. This blind spot began to clear around 2010. As sequencing costs plummeted, researchers gained the ability to extract high-quality genomes directly from ancient bones. This technology bypassed the genetic clutter of the modern world. Instead of looking at the end result of history, scientists could examine direct snapshots of populations from different points in time. We've gone from sequencing a single ancient genome a decade ago to analyzing thousands of individuals today. This scale allows us to see populations in the exact act of migrating and mingling across continents. By providing a direct look at the past, ancient DNA reveals a history defined not by isolation, but by layers of constant recurring mixture. These mixtures reach back 50,000 years to the first modern human expansion out of Africa. At the time, researchers believed our ancestors simply replaced older species like the Neanderthals. But when the first ancient Neanderthal genome was sequenced and compared to people today, the data showed a statistically significant excess of shared genetic variations. Modern humans and Neanderthals had interbred. 1,000 years ago, Western Eurasia was divided among 4 distinct populations of farmers and hunter-gatherers, groups as genetically different from each other as major continental populations are today. 5,000 years ago, a group known as the Yamnaya disrupted this landscape. They were mobile pastoralists from the steppes who combined the horse with the wheeled wagon. The Yamnaya triggered a massive migration, sweeping westward. Genetic data shows a physical movement of people rapidly overrunning existing groups, rather than a slow adoption of culture. The data from Britain is the most striking. Within centuries of the steppe migration, 90% of the ancestry of the people who built these monuments was replaced by the incoming groups. This turnover aligns with the rapid spread of the Bell Beaker culture across the continent. Ancient DNA confirms this wasn't just a change in pottery styles, it was the arrival of new people carrying the technology with them. In Iberia, the interaction was even more unequal. While the overall ancestry was a 40-60 mix, there was a complete replacement of the local male Y chromosome, suggesting the incoming males had preferential access to reproduction. The genetic similarity seen across Europe today is the physical result of this total population upheaval. A parallel history unfolded in South Asia. Modern populations are often divided by caste and language. Ancient DNA reveals that these demographics are built on similar layers of genetic blending. Most people in the region today are a mixture of 3 ancient sources: Iranian-related farmers, indigenous hunter-gatherers, and the same steppe pastoralists who moved into Europe. Genomes from ancient regional civilizations capture the early blending between local hunter-gatherers and farmers. This was followed centuries later by the arrival of the steppe pastoralists, who added their genetics to an already mixed population. South Asia's identity, like Europe's, was built through repeated, massive waves of continental blending. Because of the way biology reshuffles DNA every generation, no person carries a single, unadulterated lineage. Sequencing a single ancient individual provides a genetic glimpse of thousands of ancestors stretching into deep time. We are a network of those combined histories. This evidence leads to a clear biological reality. A pure, unmixed human population does not exist in the archaeological record. Mountains, oceans, and modern borders have never stopped the constant churn and collision of groups. Our modern differences are not permanent divisions. They are the most recent snapshot of a species defined by its ability to mix together.